Welcome to the Artists of Science, this time from Phuket, Thailand. We are with Professor Roberts, Nobel Laureate in Medicine, and Professor, it's a pleasure and honor being with you. It's our first meeting and you are participating in our first conference. Just for a general public, probably it is not necessarily the question, but for general public, tell us something about your field of research, your successes, very short.
So my field of research is molecular biology. I work with bacteria and I study the modification of the DNA in bacteria by a process called methylation. I've been doing this for many years now, but along the way I started working on messenger RNA in adenoviruses. These are human viruses, but they're not dangerous in any way. Most people get infected when they're very young and then they're immune for life. It vaccinates them to all intents and purposes. And in studying the messenger RNAs, we discovered that they were actually in pieces. They were not a single message. And they were coded on the genome in many different parts, and these had to be cut and spliced to produce the final messenger RNA, and that was the work for which I won the Nobel Prize. After winning the Nobel Prize, your field of research is the same? Well, I don't work on splicing anymore, so the process we discovered, the cutting and splicing, I stopped working in that field about 6-7 years after I made the discovery, and I've been working on DNA methylation in bacteria pretty much ever since.
And for the general public to understand, this serves what exactly?
Well, so methylation in bacteria affects gene expression. It's a part of a system called a restriction modification system that protects bacteria from phages and stops them from growing. But what we know is that there are many methylases in bacteria that are not part of restriction modification systems. They're doing something else. And for most of them, we don't yet know what they do. So plenty of discoveries to be made, which is a field I like to work in. And the nice thing is there are not a lot of people working in the field. So I like that too.
So you are a biochemist. Before, in pre-med, you're a biochemist.
No, so I started life as an organic chemist.
Organic chemist.
So when I was very young, I thought I was going to be a mathematician because I just love mathematics. Oh. And then I read a book about how I became a detective, a regular police detective, and I thought that sounded pretty good, and I thought that's what I was going to do. And then my father bought me a chemistry set, and I discovered chemistry and figured that was pretty good.
This was my question.
That took me all the way through to university, and I get to university, I'm doing my PhD. I had all the results I needed for my PhD in about a year, but in England you have to do 3 years. And so I started playing around with things, and I did a lot of reading in the library and discovered something called molecular biology and decided I was going to do that instead. And so I moved to Harvard as a postdoc, got a job down at Cold Spring Harbor doing molecular biology where I made the splicing discovery, and I do bioinformatics. So I'm a detective, I find stuff all the time. I do bioinformatics, which is all with computers, mathematics, so that's come into play. And the organic chemistry, well, life is organic chemistry. So everything I've ever been interested in is kind of all melded together into my current research career, which is very nice.
So you touched actually your reply to my question that I was planning to ask.
Oh, sorry.
That's okay. So I was planning to ask that question. So we know the story and why you ended up in your field of research. Basically, you responded to 2 of my questions that would have been asked. Now, doctor in medicine, I mean a medical doctor, MD, what's fascinating about the profession compared to other professions? Why a young generation need to go in this field?
Well, I'm not an MD.
Oh, you're not an MD.
No,
you're a biochemist.
I'm a PhD.
Oh, you're a PhD, I understand,
Totally
but not
a
MD.
PhD.
Correct.
I think people go into medicine, though, for a variety of reasons, but the best reason is because they're very compassionate people and they want to help people. And I think you will find that pretty much all of the good doctors, they're interested in helping people. They are not in it for the money, they're not in it for anything other than to try to help other humans to have a better life and to survive and overcome illness.
Is medicine a passion?
For me, no.
What's your
For
passion?
many people it is.
What is your passion?
My passion is really, I would say, bioinformatics more than anything else. That is looking at DNA sequences and RNA sequences, protein sequences, and try to figure out what they do and how they make cells, how they make life work. So that's what I like to do.
And why is not a passion medicine for you?
I think if you really wanted to make medicine a passion, you would have to go and deal with patients. And the kinds of things you do is to apply a lot of different techniques, but you don't make many discoveries as a doctor. I think it's quite difficult to make discoveries as a doctor.
That's true.
I like making discoveries.
Correct.
You know, I wake up in the
Correct.
morning, get onto my computer, I will pretty much guarantee that within the first hour, I will see something that no one's ever seen before. And you can't beat that. It's
This
fantastic.
is innovation.
Yeah, it's lovely.
Does it mean that medicine is a routine?
Well,
Kind
a
of.
lot of medicine ends up being routine, yes.
Correct.
So there are some people who do try to advance things, but usually they do it by collaborating with other people so maybe someone's come up, found a good drug, and then the clinician says, well, we could try it for this, we could try it for that. And so the collaborative nature of medicine and, say, pharmaceuticals or biochemistry, this is what can make discoveries in medicine. That doesn't happen all the time. I make a discovery every day. You can't
Every
do
day?
it every day, yeah.
In terms of?
Just I see something that no one's ever seen before. Doesn't mean it's big, doesn't mean
I
it's
know, that's
anything
okay,
terribly
it's
important.
new, it's new.
Small ones are good
But
too.
in what?
Anything new I
In
love.
what aspect?
Bacteria.
Bacteria.
Looking at bacterial methylation, yeah.
And the new aspect is in their fighting against them, or properties of bacteria, or characteristics, or?
So my passion would be, and what I'm hoping is that we will understand how a bacterium works before I die. So I want to know how they work, right? That's my passion.
That's the passion.
Yeah.
And the goal.
The goal, yeah.
And the goal. Is that goal difficult, you think?
Oh yes, yes.
And it will.
Life is very complicated. Okay, I mean, one of the reasons I don't work on eukaryotes, on humans, we're so complicated I couldn't hope to make progress. But bacteria, they're in principle pretty small, nice and compact. They don't do a lot of very, very difficult things that we know about, but maybe they are doing things that we don't know about. There's certainly plenty of things that we don't know about. I mean, if I take the DNA of a typical bacterium, if we're lucky, maybe half of the DNA, we know what it's doing. We know what genes are there, we know what RNAs are being made, what proteins. The other half we don't know.
Correct.
And so that's fun. It is nice to work in an area where people don't know that much, because then you make discoveries.
So, discovery and new ideas and new development. And then at the same time, you are an entrepreneur.
Yes.
You have created your own company a long time ago, which has been done, which is successful today.
Yeah,
So,
very successful.
how this related to each other?
Well, so the company was set up. The person who started it, Don Cohn, he'd been an assistant professor, then an associate professor at Harvard Medical School.
Yeah.
And he was in one of these departments where they just don't give tenure. So at the time, Harvard never liked to give tenure to anybody. And so he said, well, screw this. This is no way to treat academics. I'm going to start a company. I'm going to make some money, and I'm going to use that money to support research. And in this way, I'm going to set up a very good research lab, and I'm going to create Harvard and NIH all in one place. and we're
So
going to call it New
basically
England
you're
Biolabs. And I had a similar idea when I was at Cold Spring Harbor. I was discovering these enzymes that we really needed. These formed the basis of the biotechnology industry. And I talked to Jim Watson, who was director at the time. I said, you know, let's start a company. Let's sell these enzymes and use the profits to support research at Cold Spring Harbor. He said, no, you can't make money that way. It's kind of dirty to be associated with industry. He didn't want to do it. And so I discovered Don Combe. I was led to Don Combe and said, you know, I hear you're interested in something like this. And he said, yes, come up, talk to me. Went up, talked to him, and I took bacterial strains for some 20, 30 restriction enzymes. And he started making them and selling them immediately. No external source of funds. He used his own money in order to start the company. And in the first year of selling restriction enzymes, he made $200,000 back in 1975. And that provided everything we needed for the company. We've never had to borrow money.
You are selling enzymes to whom?
To researchers.
To researchers?
Yeah, researchers in academic labs, in commercial labs. The enzymes, for instance, 3 of the enzymes that are necessary to make the Moderna vaccine, we made them. We sold them to Moderna. So that's been very profitable. So coronavirus has been terrific for us because there's been a great demand for the kinds of reagents that we make. So it's been good.
Yeah.
Yes, right.
So, I mean, you like the entrepreneur spirit more than research spirit?
No, I like the research.
I like the research,
Yes.
but you use the
entrepreneurship
for the research.
Yeah, I mean, you know, I saw that here was a need, that the country, the researchers, they needed this. We were giving them all our enzymes away.
Yeah.
You know, we had this pile of enzymes down at Cold Spring Harbor. People would come to meetings and they'd come with tubes of DNA and they'd say, oh, do you have an enzyme that will cut my DNA? And they'd find one, they say, great. And then we'd give them some enzyme and then they were happy. They could go away and do their research. But it was clear there was a business here. We didn't want to spend all our time making them to give them away to other people. So.
And asking for funding from other agencies that might not understand you.
Yeah.
So you are funding your own research.
Yeah. So anyway, when I
good.
first got tied up with Don, I was offered a partnership in the company, but I didn't want that. So I became their chief consultant and spent the rest of the time at Cold Spring Harbor until 1992 when I left, and regretted not leaving earlier, to be honest. Because life at Cold Spring Harbor had gotten to be quite difficult. I'd become assistant director, which meant I had to interact with Jim Watson constantly, and he's not an easy person to deal with. When I got to Biolabs, they fund my research. I don't have to write grants. I've got a small research group that I wanted that they paid for, and I could do what I want there.
Perfect.
So now I have an ideal situation. I have all the funding I need to do my work, and the company loves it, and they let me come to meetings like this and talk to you.
Perfect. This is a very good combination, actually. It's rare. Because as I said, sometimes discoveries by Nobel laureates are not even patented by themselves, they're patented by the others. They profit from their discoveries.
Yes, well, fortunately for my discovery, there was nothing that one would obviously patent, so that was fine. With the restriction enzymes, I deliberately did not patent anything. We didn't want, because I thought this was going to slow research down. And so it was only after about, some 10, 11 years when the first 2 companies, competitors of ours, patented the first restriction enzymes. And prior to that, I'd been able to stop a number of companies from patenting. I said, we're not going to do it, and so why don't you? And so they didn't. No one did it. And then these 2 companies did. And so then we felt, well, we had to start patenting everything, which we did. And these companies were stupid. They both went out of business very quickly because they didn't have the good products that people needed. We had the good products. We were the first in line. First to market is incredibly important. And we now have still the bulk of the restriction enzyme business.
Sure.
And we've had it since 1975.
But the way you speak, it's entrepreneur spirit, the business model spirit, on top of the research.
Yeah. But
it's become your second nature, although you love research.
Yes. I have no interest in the business side of things, really.
Yeah.
I don't care for money. I have no little interest in money.
That's a good idea.
But I love research, and I have enough money to have a nice life, so that's all I need.
So, a scientist all your life in science. What about the family? Is this in science? Your wife, children?
Yes. So I have two marriages. I'm on my second marriage. I have 4 children. I think the children probably didn't get as good a father as they deserved, just because I'm a scientist. And I think one of the difficult things, if you really pursue science, it is quite difficult to provide the amount of time that you really should put in.
That's what I mentioned in my
difficult.
plenary.
Yeah.
In the opening presentation, you were there.
Yep. So that's why you have to find a wife who is somewhat sympathetic to your needs.
Starting since for Alfred Nobel.
Yes, right.
You saw that presentation.
Oh yes, I did.
That's it's actual today.
Right.
And you are also confirming that.
Yeah. But I mean, I'll give you an example of the difficulties. So in 1976, I started learning how to program computers, and we were writing programs, and I was writing them. And so one day, I just finished debugging a program, and I looked at the clock. It was 8 o'clock in the morning. I'd worked all night and didn't even realize it. And then I had to go home and explain to my wife, well, you know, I was in the lab all night. I was not off dallying with someone. And then 2 days later, the same thing happened. And I decided I had to stop programming at that point because it was obsessive. I couldn't stop. It was such fun.
Correct.
And so I hired a programmer. I now have 2 programmers who work for me. So.
That's interesting. Very, very interesting.
Yeah.
So we're on the same page in many directions. You saw our presentation. Well, what's the most difficult position when you are a researcher to do, and which is the easiest decision you can do?
But in terms of what?
In general.
But in terms of the research or in terms of life?
I didn't mean live, but since you went there, tell me both.
Well, I mean, I think in research, the most difficult decision is always to know when to stop doing something because it's failed. Okay. So I'm a great believer in failure. I think failure is when you learn. And it's very important to do a post-mortem after you fail to find out why. And this was how we discovered splicing. So our experiments weren't working, and we wanted to find out why, and it was because nature was trying to tell us something. It was telling us that the way we thought RNA was being made in higher organisms, which we knew was being made in bacteria, but it was different. In higher organisms, it was all being made differently. And so, you know, failure this time was good. But sometimes you fail, something doesn't work, and at some point you have to decide when you continue
unnecessarily.
Yes, right. And you
so
know,
this
usually
is an easy decision. This is a difficult decision.
No, it's a difficult decision. It's always a difficult decision.
Okay.
So after we discovered splicing, we wanted to really see if we could set it up in the test tube. Could we set up a test tube system to do it? We tried quite hard, couldn't get anything really to work, but a good friend of mine at Harvard did come up with a good way of doing it. It turned out he was using a different RNA polymerase to make the messenger RNA in vitro, and we just picked the wrong one. And so he went ahead, and so eventually we decided we had to stop.
What is an easy decision in research?
An easy decision? Well, an easy decision is when you suddenly have a clear view of something, of maybe an experiment that should be done, or maybe a clear view of how something might be working, and then you do the experiment to prove that that is true. Those are the relatively easy decisions to make.
And if you would go back 30 years or whatever, would you have chosen the same path?
Well,
suppose you are in high school, you have to choose the path.
I mean, I don't think I chose my path as much as opportunity came my way. I got fascinated by something, and so I followed through on it.
Okay.
I mean, you know, one interesting thing is when I was 17, we had to take A-level exams in England, and this determined which universities you could go to. So I did chemistry, math, physics, and I failed physics. I didn't like physics, I didn't like the teacher, and I didn't succeed. And I thought I was going to get thrown out of school because I'd also spent a good part of that year not going to school. But I'd become very good at playing billiards and snooker. And I went to audition to be a professional snooker player because I thought I was going to get thrown out of school. And I went through the process, but then the headmaster decided he wasn't going to throw me out of school. So I've always felt I missed, my opportunity to be really famous. I could have been world champion at snooker.
So
I
this
was
is
very
kind
good
of regret?
Sometimes a little bit, yeah.
So that was my other question, you responded. You are always faster.
Oh, sorry.
That's okay, no, it's okay. It is saving our time.
But I learnt, I think, what I feel is probably the most important lesson of my life playing snooker. I was in a tournament. I was very good. I was the West of England junior champion, and I was in a tournament, play a shot, and missed the ball that I was trying to pot, but got another one by accident. And then the next shot I made, I muffed it. I just didn't get anywhere. And afterwards, an old man came up to me. I think I was 17 at the time. He must have been in his 80s. He comes up to me and he says, you could be a really good snooker player, but the one thing you have to learn is that when you have a lucky break, when something lucky happens, you've got to concentrate twice as hard on the next shot to make sure you don't waste that luck. And I think this is one of the most important lessons that anybody can learn in life. It really is important when something lucky happens, make the most of it. Don't let that lucky break go away. And a lot of people do. A lot of people feel guilty because they had the luck, but everybody has luck. Absolutely everybody. And you have to take advantage of it, and the smart people take advantage of it.
Perfect, well said. Just the last question. What are your hobbies besides science, besides research?
I play croquet.
Croquet?
The game of croquet.
Okay.
This is how I spent my Nobel Prize money, was putting a croquet lawn in front of my house. Something that a writer in the Scientific American said, well, some people just waste their money by putting in silly things like croquet lawns. But for me, it's been great. We got it finished it's a professional lawn, finished in 1994, and I play as often as I can.
Perfect.
I love it.
Professor Roberts, it was enlightening discussion and conversation. We should have more like this.
Sure.
We always have less time than we need.
But maybe I can ask you the questions.
Sure. Next time, yes, for sure. By the way, you are invited to our next meetings, and I'm going to send you an invitation.
Okay.
But this is really enlightenment.
Sure. Yeah.
Thanks a lot, and you discovered a part of yours that I was not aware. It's my fault. The entrepreneurship.
No problem.
Next time we'll speak more about that.
Sure.
Thanks a lot.
Thank you.
An artist of science.
And thanks for inviting me to the meeting.
Thank you, it's a pleasure.